Hello students, today's topic is pollen-pistil interaction, how the pollen will interact with the pistil, what will be the fate of pollen when it falls onto the stigma. Pollen-pistil interaction can be defined as transfer of pollen grains of one plant to the pistil, stigma of the same or different plant, which can be either self or cross-pollinated. Pollination is the first step of pollen-pistil interaction, followed by the pollen addition to the stigma. Pollen grains are deposited on the stigma either by self-pollination or by pollinating agents, biotic or abiotic. This unique feature brings about pollen-pistil interaction between the male gametophyte and the pollen grains with the massive sporophytic tissue. For pollination to occur, the pistil needs to recognize the pollen grain of the same species. Pollen-pistil interaction is not a small process. One needs to understand the process of pollen-pistil interaction, which ultimately results in successful fertilization. What are the various steps? First step is landing of pollen grain onto the pistil. If the pollen is compatible, the pistil accepts it, while incompatible ones are rejected. If the pollen grain is accepted by the female plant, the grain forms pollen tubes and releases its contents and chemicals. Pollen germination involves the creation of pollen tubes which allow pollen grains to release their contents. Pollen tube develops into the ovary via the style of the pistil. Male gametes enter into the ovule and subsequently into the synergid. In short, it begins with pollination and ends with fertilization. In general, pollination is followed by 4 steps. First, landing on the stigma, then it's followed by 4 steps. What is that? Pollen recognition by the stigma, which is followed by the germination in the stigma, within the stigma. Then it's pollen tube passage through the style till it reaches the micropyle. Then it's entry inside the embryo sac through one of the synergids and its release, and then the discharge in the synergid of the embryo sac, and later double fertilization. So in total, various steps taken by the pollen. First, pollination. Second, addition onto the surface of the stigma. Pollen hydration, pollen germination, pollen tube entry in the stigma. It's followed by pollen tube growth through the style, then pollen tube entry into the embryo sac, then it will be followed by double fertilization. All these events form a part of pollen-pistil interaction and are called programmic phase. Now let us talk about the structure of pistil. What is pistil? Actually, the female reproductive part of the flower is called gynoecium and the individual member is called pistil. It comprises of variously shaped structure at the top of the style, that is stigma, long filamentous tube, the style, and lower bulbous portion, ovary. Whatever the structure of stigma, it is the place where pollen lands, germinates, and entry of pollen into the pollen tube into the ovary, into the ovule, and then into the embryo sac for fertilization takes place. Structure of stigma, which is the receptive spot where the pollen lands, is of various types, covered with cuticle and proteinaceous lipolidal layers called pellicle. You can see the shape, the diagram. It's differentiated, filiform, not differentiated, club-shaped, conical, disc-shaped, hairy, feathery, comb-like, lobed, or it can be beaked, or it can be bifid, trifid, pentafid. Its surface is glandular because of the secretions it produces or not. So it's of 2 types, wet or dry. The composition of secretions produced by hypodermal cells of the stigma is rich in lipids, proteins, glycoproteins, various types of enzymes, amino acids, calcium ions, and phenols. Pollen land on the stigmatic surface and get hydrated. So the stigma are of 2 types, wet and dry. Now let us first talk about the structure of wet stigma. Main function is to provide pollen with water necessary for germination. Due to exudates rich in lipids, proteins, glycoproteins, amino acids, various types of enzymes, free sugars, phenols, polysaccharides, secreted by the stigmatic surface, accumulation in interspaces form interconnected systems and loosen up. Continuous secretion results in enlargement of intercellular spaces. cuticle pellicle layer releases exudates which make the stigma wet. For example, Petunia. Structure of dry stigma. No exudates released as cuticle is discontinuous. Pactocellulosic layer differentiated into many layers. Pellicle and cuticle present. Pellicle shows ATPase and carbonic anhydrase activity. Since these stigmas do not secrete exudates, they are called dry stigmas. For example, Linum species and Gossypium species. Function of stigma. Pollen-pistil interaction. Stigmatic exudates appear as droplets and serve as nutrient source for both pollinating agents as well as it aids in pollen germination. Lipolydyl component of the exudate prevents evaporation. This keeps the stigma wet, while phenolics and protease inhibitors protect stigma against insect and pathogen. Pellicle exudate acts as an adhesive. Just after stigma, let us study the structure of style. Just below the stigma is tubular structure which connects stigma to ovary. It's called style. It is mainly of three types: solid, hollow, semi-solid. Solid type, central strand made up of transmitting tissue, elongated plasmodesma, interconnected cells, proteinaceous secretion containing proteins, glycoproteins, and lipids surround the cells. Cheng et al. 1993 reported presence of number of transmitting proline-rich proteins in the intercellular matrix. Cells contain many mitochondria, active dictyosomes, rough endoplasmic reticulum, blastates, and ribosomes. It is through this cellular matrix the pollen tube moves. Example mostly dicots. We're talking about structure of style. A holotype central core has one or more canals which are equivalent to the number of carpels. The stylar canal is bordered by one or few layers of glandular cells, the canal cells, and covered by cuticle, which remain filled. These canals remain filled with fluid. Example, crocus, amaryllis. And semi-solid one, intermediate between two. Example, cactus. After landing on the stigmatic surface, pollen grain adheres to it, to the papilla, and imbibes water, swells up, ruptures along the germ pore, and in time comes out in the form of pollen tube. Then the pollen tube traverses the surface of stigmatic papilla or passes through the cellulosic pectic layer of the papilla wall and finally reaches the base of the papilla, from where it migrates through the intercellular spaces of the stigmatic tissue, and further growth of pollen tube depends upon the structural nature of the style. After germination, the pollen tube elongates, it passes through the style, and it actually has to reach the embryo sac. So fully developed pollen tube has compartmentalized appearance due to the presence of callus plug. It's in the form of a ring-like structure which appears, and it grows inwards towards the center and finally seal the tube. These plugs prevent the backflow of cytoplasm and nuclei. Meanwhile, gametogenesis is over. Microgametogenesis, 2 male gametes and 1 tube nucleus moves towards the tip of the pollen tube, and plugging takes place so that the backflow of the cytoplasm and nuclei is prevented. Now let's talk about pollen tube guidance. The pollen tube, after traversing style, reaches the basal swollen portion of the pistil, the ovary, and later into the embryo sac. There are three methods of entry. Porogamy, that is typical. If it makes an entry through the micropyle, if it makes an entry through the callus, we call it callosogamy, as the example is casuarina. And if it makes an entry through the integuments, as in case of cucurbita, it's called mesogamy. Whatever the mode of entry of pollen tube into the embryo sac, the tube nucleus guides the pollen tube till it touches the embryo sac, the female gametophyte, and then the tube nucleus degenerates. Entry of pollen tube inside embryo sac takes place through one of the synergids, and it reaches the cytoplasm of the synergid. Electron microscopic studies have revealed that pollen tube enters through the apical region of the filiform apparatus, grows through it, and reaches the cytoplasm of the synergid. The pollen tube discharges its contents into the synergid, but does not grow beyond it into the embryo sac. Usually, the penetrated synergid gets destroyed due to penetration of pollen tube, whereas the other synergid degenerates after some time. The contents of the pollen tube released inside the synergids will automatically be transferred into the embryo sac, and after this, double fertilization will take place, which is in another video. Thank you for patiently listening. Subscribe it, like it, and share it. Thank you.